What's that?
We're gonna have to grade by the
Probably.
Probably. Is it bad? Are you
No.
gonna have to put a big curve?
No, I'm not. They're actually pretty good.
Can you put a curve anyway?
Yeah, I've been checking.
Whenever you get the test pack, just leave it back.
Just leave it back up there. What, oh, you don't want to see yours? Okay.
It can't be that bad. How big was the curve?
I haven't decided yet because I'm not quite done.
Oh.
You estimate.
Yeah.
No. No, it won't
What
be.
do you consider a big curve? Yeah, like last test, I was not expecting us to have an 8-point curve, and then you were like 8-point curve, and I was like,
Somewhere around there, I guess.
oh,
So
that's
somewhere around
good.
8 points? Oh.
Did everybody do
But-
the thing?
Oh yeah, that's what I wanted to talk to y'all about, but I'm waiting until the time.
Send me your Moodle password.
Lecture got
What
it.
day? What?
Lecture, yeah, lecture got it. Lecture got it. We have 80%.
We have to do it for lab too?
Yeah,
No, wait. For the bonus.
yeah.
Oh.
Every single one of my classes, if you do it, you get 5 bonus points on the final exam.
Do them anyway.
So wait, so if there's 75% on the lab, you also get 5
I think they
points on
close
the lab?
Yes.
Oh, all right. I'll do this.
No, it
Yes,
doesn't.
please do the lab.
But it doesn't.
Yeah, they're like, we're not in lab, we don't care.
Wait. Does lab have 70%?
No, lab does not. Lab is only at like 51% or something
oh my
like
God.
that.
I didn't realize this.
Lab is low. Yeah. So like,
I was gonna do it. But I was waiting.
you were just waiting. Well, so if you have not done it, the course evaluation for lecture is at 80%. That does not mean that if you haven't done it, don't do it. Please do it. I want your feedback. It helps me. Tell me what was good. Tell me what you think you might like to change, whatever it is. Be honest, but be positive criticism, right? But then lab is only at 51%. So if you have not done it for lab, please do it for lab because lab gets 5 bonus points on their final exam as well. So what's that?
Please, I'll do it.
Please. Everybody's begging in class. Please do it. Please do it. So, but no, see, I had it on my sticky note with a smiley face, right? Because everybody likes to get smiley faces. I did.
Yeah.
I did.
She gave me 8 bags of Ziploc bags to bring home.
No.
They were sandwich size. They
Oh,
were
it was-
sandwich size, but still.
That's pretty big.
Yeah. All right. So, what do you call a deer with no eyes?
No idea.
No idea. No idea. No idea. Yeah.
Really close, but
All right. Anyway. What? Oh, I have another good one. Y'all guess that one. All right. Let me tell you another one because y'all guess that one.
That one was too easy.
What kind of music does a fish play on the piano? Scales. My son told that to his piano teacher on Thursday. We have piano Thursday night. So he was so excited that he had a music joke to tell his piano teacher. It was awesome. Okay, so the last thing we were talking about was, right, everything that has to happen in the electron transport system. So what I want to do is, before we even go into section 1, I want to tell you what we're going to learn in section 1. So normally, right, I have a little handout for y'all and we fill it out together. I want you to take a sheet of paper out. If you don't have a sheet of paper, who needs a sheet of paper? I got paper. Who needs it? Ooh, I'm getting caught on the desk. Y'all need one? Yes, no? Yeah, no. Anybody else in this row? Don't write it on your iPad, whatever, write it whatever. Doesn't matter to me. But like, this is or anybody else? No? Okay. Anybody needs one up here? Okay. So there are 4 big things that we're going to get through in section 2, right? So section 2 is the ETC, right? In this figure up here, what's the ETC? Out of all this stuff, what's the electron transport chain? This stuff, right? There are other stuff that comes after it, but section 2, we're just going to focus on the electron transport chain, okay? So we're going to look at the 4 functional Components, components of the ETC. Okay, I'm betting you can guess number one. The first functional component are the complexes, right? Those big multi-subunit complexes. And the ones that we're going to work on are 1, 3, and 4. Why isn't 2 in there? This is a multi-subunit complex. 4 was a what? You just said it. 2, I'm sorry, I said 4. 2 is an enzyme within the citrate cycle, right? So it's not really a complex. We call it complex 2, but it's not really a complex, okay? So that's one thing I think is really important to kind of start out with. Not end with, but start out with. Okay, now, in order for electrons to move within these complexes, between these complexes, what are you going to have? Oxidation reduction happening, but that's true in every single part of this. But like, you got to think complex 1 is something that's embedded in the membrane. Complex 2 is something embedded. Complex 3 is something embedded, right? So how do I get my electrons from these big complexes from one to the next? What do you have to have? Mobile carrier proteins, right? Okay, things that can move. So one of them is coenzyme Q, right? What's another word for that? Same thing as ubiquinone. U-I-N-O-N-E, ubiquinone. What's that? Okay, so these are small, they're hydrophobic, and they're mobile electron carriers. So there are multiple mobile electron carriers. Ubiquinone specifically will transport electrons from where to where? From 1 to 3. What else? Gosh, one too many lines. And there are some other things that Q carries, and I'm going to come back to that, okay? So this is not an extensive list, but we'll get there. Okay. So then, if you're not a complex, you're an enzyme, right? So you are membrane associated enzymes. And these membrane associated enzymes are FAD containing. Do you remember how we've talked about lots of different enzymes can have cofactors and things that are required in order for it to happen? FAD is one of them. We're going to talk a lot about FAD today. So that's kind of just be aware, and it'll make sense later. So there are 3 different membrane associated enzymes within the electron transport chain. What's the one that you already know? Complex what? Complex 2, which is succinate, succinate dehydrogenase. That's one you know. Now, the other 2 you don't know, but what happens when you go on a low carb diet? Where do you get your energy? From fats. So you have triacylglycerides, which are a glycerol and 3 fatty acids, right? So what happens is we want to convert those fatty acids into acetyl-CoA, because then the acetyl-CoA can enter what? Citrate cycle. But when you make that conversion, you are actually going to release some electrons. So that conversion from fatty acid to acetyl-CoA that can then go to citrate, this right here is going to release some electrons. And those electrons right there are going to be picked up by the ETFQ oxioreductase. OXIDOUCUCTASE. The ETFQ oxioreductase. So these are the electrons that get transferred there, which we haven't really talked about this yet, but I feel like it's important to get a good picture beforehand before we get into the nitty gritty. ETF-Q. I know it's something you haven't seen before. There's one more that you haven't seen before. And sometimes we need a shuttle system in order to transport those energy carrying molecules. And so we have one more enzyme that will shuttle those carrier molecules around. And so that is glycerol 3 phosphate dehydrogenase, right? And you can just put in parentheses shuttle system. And we'll come back to that. So you really only know right now complex 2, but we'll talk about these other 2. So what's common in all three of these enzymes is that all three pick up electrons and give them to, where are we going to give them? You know complex 2, where do the electrons go? What? To 3. Right, that's the kind of common thing. So they're going to go to Q, Q goes to 3. Okay, all right, now we have one more, and I keep not seeing my next page. There it is, okay. Number 3. What did we leave out? This is 4? 4. I said 4. I said 4 different things, didn't I? 4, thank you. What did we leave out? Cytochrome C. So what does cytochrome C do? Carries electrons. Now, cytochrome C does something special with those electrons. It only carries them one at a time. It's really important. From where? 3 to 4. Okay. All right. I want us to try something.
It's from 3 to 4.
Yeah, I'm sorry, from 3 to 4. I want us to try something. I want us to try to draw what we just wrote. Okay? Sometimes turning it into a visualization really helps. When you're studying, this is a really good study tool, really good study tool. Okay, so let's start with complex 1, right? What happens with complex 1? What's going to come in? NADH is going to come in. It's going to drop off its electrons and make plus H plus, right? Okay. When that happens, what's my result? Eventually, 4 hydrogens, 4 H plus, are going to translocate. So that means that I'm embedded in a membrane here, right? So this is my, and I have another membrane here, right? This is my outer membrane. This is my inner membrane. And what do we call down here? matrix. With me so far? Okay. Now, where are those electrons going to go? They can't go straight to 3. Something's got to carry it to 3. What's going to carry it to 3? Ubiquinone or Q. So Q's kind of a mobile electron carrier. What? Yeah, a mobile electron carrier, right? And so now we can take these electrons and we can send them to complex what? 3, but I made a big gap there, so that should be a good sign to you, right? Okay, so Q can take those, but Q doesn't stay Q. If it's picked up electrons, what does it have to turn into? What happens when you pick up electrons when you're over here? When you go from NAD plus and you pick up electrons, you become NADH, right? So we gotta change some hydrogen. So actually we're gonna be QH2. Now we're at QH2, now we can drop off at 3. Okay, what else? I didn't really draw this too well, but my screen's just not big enough. Okay, so let's draw another complex. What is this complex? Complex 2, right? Okay, so what I actually did, what you actually have to kind of keep in mind is that there were 2 hydrogen ions that got picked up by Q, right? So Q's actually pulling 2 hydrogen ions along with it. Okay, we pumped 4 and we're basically recruited 2. Okay, so now what happens at 2? FADH drops off FADH2 to make FAD, right? Okay, so now 2 can give these electrons to Q, right? But in essence, we've bypassed 1. Yes? Okay, so these are 2 different ways that Q can pick up electrons, but those aren't the only ways. There are 2 more ways that Q can pick up electrons. Go up to your list up above. Where else can Q get electrons from? We'll call this, yeah, there's like 2 other membrane bound things, right? So we'll do them like this. Okay, so one of them was what? Glycerol 3-phosphate dehydrogenase. This was the shuttle, right? And then what was our other one? It's the fatty acid one, right? The ETF, I could write today. ETF, Q, oxio. Oh my gosh. ASE, and this is for fatty acid oxidation, right? ION, okay. So where do they give their electrons to? Q. Q converts to QH2, takes those electrons to complex 3, right? Y'all doing okay? Okay, tell me, like pause, Dr. B. We need like 5 seconds. It's okay. If you don't tell me, I will just keep going. Did I just draw 2 hydrogens? What? These? Yeah. What do you mean? Did you just draw them? No, they've been up. These are these 2 hydrogens. These 2 are the same. Yeah? Okay, I just missed that. That's okay. It's okay. Because we said 4 are pumped and 2 are recruited. Okay, so now we're going to bring those electrons to complex 3. When they get to complex 3 is going to do what? When we transfer those electrons, it's going to pump 4 because we have 2 here, right? And we have these 2 here. We're going to pump 4. Right? And I go this way. Okay, now what's going to happen? Cytochrome C is going to pick up those electrons and going to transport them where? Complex 4. But it's not going to pick them up 2 at a time like Q did. It's going to go 1 at a time, right? So you're going to say 2 times 1 electron. It's a good way to say it. What happens here? So this one is not 4 because we're at the very end. Think about like you're losing energy as you go. So here we were able to recruit 4 plus 2. We were able to recruit 6. Then we were able to pump an additional 4 along with those 2 recruited. So now this one's even less. This one's only 2. So we're only going to be able to pump, I can move this, 2 hydrogen ions this way. What else happens that's really important here at the end? We make water from 2 hydrogen ions plus 1/2 O2, which doesn't really exist, but that's okay. And we're going to make H2O. So that's the end of just the electron transport chain, right? There's more that happens, we know that, but this is what we're going to focus on today. Okay, now you have a good picture, you know where we're going, you know the plan. It only took like half the class. Really good sports. Okay, you ready? You need a second? You just in shock? Okay, take a second. And all this is in your book, but like to build it yourself helps you to retain it, helps you to understand it. So that's a good study tool for forever and a day. This isn't really chemistry. Yes, it's chemistry. Everything's chemistry. It's a beautiful combination of biology and chemistry. Okay, so where is oxidation occurring in this model? It's occurring in all the complexes, right? Oxidation, we have oxidation reduction throughout the whole thing, right? So where does it start? It starts either in complex 1 or complex 2. So if you're in complex 1, what's being oxidized? NADH is being oxidized, right? On what side of the membrane? The matrix side, right? Okay, so like if you ask yourself these questions while you're looking at your diagram, it's going to help you too. All right, and then same thing's true for FADH2, right? You're still on the matrix side. Because what happened? How do you know that NADH and FADH2 are on the matrix, in the matrix? Citrate cycle takes place in the mitochondrial matrix, right? We had to pump, we took our pyruvate, we converted it to acetyl-CoA, we push our acetyl-CoA into the mitochondrial matrix, so things that are generated in the citrate cycle are automatically in the mitochondrial matrix. Okay, it's good to know where you're going. All right, so now, like, there's so much stuff. Y'all, I have to tell y'all, this is a funny, quick funny story. Y'all like my funny stories, right? So, my mom is a retired. She was the director of the Barataria Terrebonne National Estuary Program right here on campus. Y'all know BitNet. Anyway, so she's given a talk on a virtual conference. And so she calls me, she's like, I have to record this talk. I don't know what to do. And I was like, it's okay, Mom, come over, I'll help you out, right? It was a 9-minute talk. And she had it done. She knew what she wanted to say, all that kind of stuff. It took us about 2 hours. She was like, I have a deep appreciation for everything that teachers are doing right now. I said, okay.
You have to do it for genetics. You have to do that like a Zoom project.
Uh-huh.
And it was like, my part was like 2 minutes.
How long did it take you to do 2 minutes? A long
2
time.
hours.
Yeah. So yeah, so it's all this technology stuff, right? That made me think of it. Okay, so we already talked about this, right? NADH oxidation occurs where? Complex 1, and it takes place on the matrix side of the inner mitochondrial membrane, right? What happens? It's 2 electrons that initiate many redox reactions. We just showed complex 1 is this blob, right? But guess what? That blob is a lot of different proteins, a lot of different peptides that are working together, right? And so you're going to have lots of oxidation reduction reactions happening within that one blob complex, okay? So eventually it's going to end with oxygen being reduced to water, right? That's the end of NADH. The alternative is that 2 electrons can enter the electron transport chain through FADH2 oxidation. Where does this start? Complex what? 2 to complex 3 to complex 4, right? So I'm just going to say 2 to 4, right? This is 1 to 4. This is 2 to 4. So that's kind of like 2 separate things, right? You're still going to end up reducing oxygen to water here, okay? So the flow of electrons, why do electrons move? Reduction potential. Reduction, it all boils down to reduction potential, right? So our lowest reduction potential is somewhere like negative 0.32, right? And we're going to go all the way to oxygen, which is a positive 0.8, 2 or 3, something like that. Volts, these are volts, right? Remember your units. Okay, so is it going to go in one jump? No, it's really like this sequential, right, hopping of one to another as we increase our potential. And we harness that loss of energy there by pumping hydrogen ions, right? Yes? Okay. Okay, so when you look at these systems, right, this is what we've already kind of said. If you start out with NADH, we're starting in complex 1. And you drop off those 2 electrons in the NADH ubiquinone oxioreductase complex. That's complex 1. That's why we just call it complex 1. Isn't that so much nicer to say? But do you have to know this? Yes, but it makes sense because the electrons are coming from NADH and they're going to ubiquinone, and it's an oxidation reduction reaction. Oh, beautiful. Beautiful. Just love it when that works out, right? So 4 electrons are pumped out. 4 electrons. 4 protons. Woo! 4 protons are pumped out of the matrix into the inner membrane space. 2 other hydrogen ions are recruited to Q because Q is being what? If we go from Q to QH2, reduced, okay? So now that we have QH2, we're actually carrying 2 electrons in the form of QH2. QH2 will bring those 2 electrons to complex 3. Once those 2 electrons are dropped off, we have release of these 2 hydrogens into the inner membrane space, and we pump 2 hydrogen ions to the inner membrane space.
So the reason why the electrons go to ubiquinone is because the hydrogens attaching to it.
Correct. I'm going to show you. I'm going to show you. I'm going to show you. So now we're at ubiquinone. We're at complex 3, right? We skipped complex 2. So we're at ubiquinone cytochrome C. So ubiquinone's on this side, cytochrome C's on this side, right? Okay, that's why we named it that. So we're gonna drop off these electrons to cytochrome C, but cytochrome C can only take them one at a time to complex 4. Once we get to complex 4, right, those 2 electrons will eventually reduce our oxygen into water, right? And at the same time, we're gonna pump 2 hydrogen ions from the matrix to the intermembrane space. Here are the other possibilities of what could happen. There are 3 other possibilities. You could have FADH2, right, that came from where? Citrate cycle, that can drop off electrons to Q. You're still going to convert Q to QH2. QH2 is still going to go to complex 3, so on and so on and so on, right? You could have fatty acids. You've gone on a low carb diet. Good for you. I couldn't. I like my carbs way too much. So here are my fatty acids, right? I'm going to produce FADH2 when I convert those fatty acids to acetyl-CoA. So that FADH2 is then take those 2 electrons from FADH2, give them to Q, make QH2, so on and so on, right? So this membrane-associated enzyme is the ETFQ oxioreductase, right? The other thing that can happen is sometimes we have FADH2, right? That we need to convert back to FAD, but we're not in the matrix. And so this is the shuttle system, the glycerol-3 phosphate dehydrogenase, that will allow us to cycle that FADH2 back to FADH. And we'll talk about why that's important later. Those are all the things we've already covered so far, right? Okay. So if I were to say A pertains to what pathway? This is the NADH pathway, right? Yeah? What's B? This is FADH2. Really, both of these are FADH2. They're just FADH2s generated, one inside the matrix, one inside basically the cytosol. And then also fatty acid oxidation. OXIDATION. Right? Okay. All right. So we know that the electron transport system is super important. What happens if you block any part of the electron transport chain? You will die. You will die. You will stop making ATP and you will die. So there are lots of different things that can do that. So rotenone is an example of a compound actually that are isolated. It can be found in plant roots, and it's actually used in some tribes to make poisonous arrows, which is kind of cool, right? Hydrogen cyanide we talked about when we talked about the Nazi gas chambers. That's what they used in their gas chambers. Carbon monoxide. Let's talk about this one real quick. Why is carbon monoxide lethal? It binds to the oxygen. It doesn't bind to the oxygen. It binds to the hemoglobin preferentially. So it actually has, sorry, y'all. It actually has a 250 times higher binding affinity to hemoglobin. So if it is present, it does not matter how much oxygen is in your bloodstream, it is going to preferentially bind it. So what do you have to do in order to get rid of carbon monoxide? You have to get away from the carbon monoxide, right? You have to decrease the concentration of carbon monoxide. Okay. Antomyosin A is another antibiotic, kind of like we talked about with oligomycin, right? But it just prevents complex 3 from working. But it doesn't matter where you inhibit. Maybe it's in the electron transport chain. Maybe it's your ATP synthase complex. And maybe it's even the ATP-ADP translocase membrane, oh my God. translocase proteins, but it doesn't matter where you inhibit, you're going to back up the rest of the pathway. Okay, so just to keep in mind how important it is. All right, so this is really, really similar to what you saw in the textbook, like in the last chapter, right? But it's flip-flopped. So be careful, right? Now at the top, we have our lowest, what? Reduction potential, right? So this is the direction of electron flow from the top of the chart down to the bottom. So what do you see? You're going from NADH to complex 1. But remember how we said there are lots of different things inside of complex 1? Well, these are some examples of things that can be oxidized and reduced in order to pass electrons along, right? So what you're going to see is those changes in reduction potential. What do they do? They increase. So as you go, they increase. And so these are just the different things within each of the complexes that the electrons get passed to. And we'll talk about them kind of in detail. But eventually we get to oxygen. Okay, so how is it that you actually can translocate hydrogen ions from the matrix to the intermembrane space, right? That's kind of like, it's kind of been, I've just kind of said it happens, right? I haven't really explained why it happens. So there are two different ways that it happens. There are redox loops and there are proton pumps. So in a redox loop, right, you have something that is carrying those electrons, right? We're going to transfer those electrons to our mobile electron carrier, turn it into like QH2, right? Bring it to the next complex. Now, those two hydrogen ions can then be passed to the intermembrane space. I didn't actually pump them, right? So that's a redox loop. It separates protons and electrons on opposite sides of the membrane. This happens in the Q cycle in complex 3, and we're going to talk about that in detail. The other way that this can happen, right? We have our electron carrying molecule, right? It binds to a mobile electron carrier, right? That picks up hydrogen ions, that's fine. Now, when we get to the next complex, what happens is we take those two hydrogen ions that were on like the QH2, right? And we're going to convert another species that was in the inner membrane into one that is now carrying those hydrogen ions. But in the process, it makes a conformational change in this protein. That conformational change actually pumps two hydrogen ions. So one is a pump and one is just a redox loop. And the details, any more details than this, they're still working on. Like this is not a completely understood system yet. It's still something we're working on, but that's a good generalization, right? Okay, so let's kind of take this pathway straight through, right? Complex 1, we know we're NADH ubiquinone oxidoreductase. So our NADH is oxidized, our coenzyme Q or ubiquinone is reduced. So this is the largest complex. You're anywhere from 15 to 40 polypeptides. Depends on what organism you're in, how complex it really is, right? But this is one that requires our flavin mononucleotide, our FMN. FMN's really similarly, let's write with a pen on the board, not good. Similar to FAD, very similar to FAD. Not quite, but similar. So FMN accepts our 2 electrons from NADH. So this is kind of, if you didn't have FMN, this whole complex wouldn't work, right? So what do we call those? What's this an example of? A prosthetic group, right? So a prosthetic group, these are things like, right, vitamins, right? We talked about that when we talked about what? What was it? Pyruvate dehydrogenase. Yep, exactly. So lipids, that was another one, right? A metal ion, right? These things are not amino acids, right? Not proteins. Okay. Now, we're in complex 1, right? So we have our NADH. We're going to give up our 2 electrons. FMN is going to pick up those 2 electrons. So those 2 electrons are in the form of what's called a hydride ion, right? So do you see the 2 dots? The 2 dots mean that these are the 2 electrons that this hydrogen has picked up. But this is just the center proton. So how many protons are in there? 1. So if I have 1 positive, 2 negatives, what's my overall charge? -1. You've probably not seen that before, huh? You have? Yes? Some, yes? Ooh, good. Awesome. All right. Okay, so then those electrons are transferred to iron sulfur centers. So these iron sulfur centers are able to carry our electrons because of the iron. Tell me about iron. We can be Fe3 plus, we can reduce to Fe2 plus, right? So this is reduce. If you are picking up, okay. All right, but what do you know about that? How many electrons did we pick up? 1 at a time, 1 at a time, right? Until we get to the very end, and then it takes 2 electrons to convert Q to QH2. Yeah. So in the process of transferring all of those electrons to all these different iron sulfur centers, what you end up doing is actually pumping 4 hydrogen ions from one side of the membrane to the other side. So this is a pump. But what about these 2 hydrogen ions? These are being picked up by Q to make QH2. So these are going to go into a loop, a redox loop, right? So this is both mechanisms. Okay, so kind of keep that in mind as we go. All right. So just to give you a little bit of a background on FMN. So these are also reduced 1 electron at a time, just like we said in the iron centers. But what happens is you look at your nitrogens, right? They're in the double bond here, and then the nitrogen over here in this double bond. In the first reaction, when it picks up an electron and a hydrogen ion, so these are the ones we're looking at, here and here. We have an intermediate, right? This intermediate only has 1 electron and bound to the hydrogen. That's why we see the 1 dot right there, right? That's where your extra electron is. Then we pick up another, and this is our fully reduced. So it picks them up 1 at a time, but it can carry 2. Okay. So where are those electrons going? They're going to coenzyme Q. Have you seen it like the health food store? CoQ10. Sit, right? No, well, you know. It's at Rouse's too, I think. All right, so coenzyme Q is our mobile electron carrier, right? Because it can move in the membrane. The other complexes are completely stuck. They're not going anywhere. So that's going to transport our electrons from complex 1 to complex 3, right? It's also the entry point of our 2 electrons from the citrate cycle, and our electrons from fatty acid oxidation, and our electrons from glycerol 3-phosphate dehydrogenase. But we already drew all that, right? Okay, so here's the place where we convert from carrying 2 electrons at a time to carrying 1 electron inside of Chrome C, right? We have to figure out a way to separate those electrons from being carried 2 at a time to 1 at a time. It's kind of complex, and it's called the Q cycle. Ask anybody in biochem 2, they don't like the Q cycle. They don't. Poor things. Okay. So ubiquinone. So how is Q going to pick up those electrons? It's the same sort of thing. What we're going to do is go through an intermediate form. So here, if you look at this, right, we have oxygen double bound to a carbon, oxygen double bound to a carbon, right? And so remember when I said CoQ10, right? So all that 10 refers to is how long the hydrocarbon tail is. There are many different forms, but this is the one that is most common in humans, right? So that's why they call it CoQ10, and that's why you find that vitamin at Rouse's or the health food store, whatever. Okay, so we go to a semi-quinone intermediate, right? So we picked up one of our electrons. Then we go through a second reaction where we pick up a second electron and 2 hydrogen ions. And so both of those oxygens now turn into hydroxyls, right? So that's why we're QH2. All right, so where are we going now? We're going to 3, but what's the other way to get to 3? From 2, so let's talk about that real quick. We're in 2. In order for 2 to pick up electrons, where are those electrons from? FADH2. We generated FADH2 when we made fumarate in the citrate cycle, right? So whenever you oxidize succinate to fumarate, you're going to make FADH2. That FADH2, guess what, is picked up by a chain of iron sulfur units. And you also have, this is another cytochrome. This is a cytochrome that's actually embedded. It actually can't move. Some cytochromes are mobile, some cytochromes are not. So this is a cytochrome, CHRO, cytochrome B560. There are many, many, many different types, right? Yes, oh, just a stretch. Like I was so excited. All right, so this is the other way, right, that we can pick up 2 electrons to Q. Did we pump any hydrogen ions? No, right? So we're not going to get quite as much ATP per FADH2 molecule as we do NADH.
So since we're not pumping any hydrogen, this is considered redox.
It's the beginning of a redox loop. Yep, yep, exactly. All right, so whether you came from complex 1 or you came from complex 2, doesn't matter, you still converted Q to QH2. So complex 3 is our ubiquinone cytochrome C oxidase. So we're going to reduce our cytochrome C, and at the same time, we're going to translocate 4 hydrogen ions. This is why we say translocate, and we don't say pump, right? Because some of them you're picking up from the redox cycle, some of them you're actually pumping. So you have to kind of be careful about that. So on this complex, on complex 3, you have a docking site for QH2, and you have a docking site for cytochrome C. So QH2 comes in on one side, cytochrome C will leave and come back from the other side, right? So there are 2 unique binding sites for ubiquinone. It's called the QP and the QN. So you can say 2 binding sites for ubiquinone. It's going to be really important why in a second. So again, our transfer of electrons is through iron sulfur clusters, just like what we were talking about before. But this time we're going to do them one at a time, right? So there are 11 different protein subunits in this complex itself, right? So when we look at this, right, here's our Q cycle. So we have our QH2, right? Our QH2 is carrying how many electrons? 2, okay? So we call this binding, right? Because remember we said over here, we have 2 binding sites, QP and QN. All right, the QP site has 2 hydrogens attached to it. So I have 2 electrons. 1 electron is going to get transferred to an iron sulfur center. The other electron is actually picked up by cytochromes. So 1 electron went straight to cytochrome C, another electron followed this path, right, is going to be rebound to the free Q. So now I have Q, that intermediate form, that only has 1 electron attached to it. So this is the QN site, okay? So now we can bring in hydrogen ions from the mitochondrial matrix and convert that intermediate Q into QH2. Now QH2 can come out and cycle again. This is how we split those electrons up, right? Because cytochrome C can only pull one at a time. So that other electron has to go somewhere and be held until we're ready for it. So that's how we split up the two. So these B's, right? Those are cytochromes, but those are immobile cytochromes. Only cytochrome C is a mobile electron carrier and can bring it to the next complex, right? So in order for this cycle to happen, to actually transport two, you're actually going to need a second QH2 molecule to bind in the QP site, right? Because then you need a second electron to convert this back to a full QH2. So it's a little convoluted, but it works. It really works. Okay. So cytochrome C, it's a really small mobile protein that associates with our inner membrane side, right? And this is the one that's responsible for converting it from 3 to 4. And it uses an iron-containing heme prosthetic group. So we've talked about hemes before, right? Just having iron in that center, what had a heme? Hemoglobin, right? So easy. So there are different types, and it's all about the additional things that come off of that heme prosthetic group that turns it into an A type, a B type, or a C type. And this is the C type. This is the type that's actually mobile within the membrane. And these are really super highly conserved, their structure and their sequence, right? Okay, so where are we going now? Complex 4, right? It's like trying to keep track of many little children going everywhere, right? Okay, so we have cytochrome C that's got our 1 electron, but we're going to do it twice so that we keep track of 2 electrons, because that's kind of what came into the system. So this complex is called the cytochrome C oxidase. What are you doing to cytochrome C? Yay, okay, good. So we're going to transfer those electrons. You're going to go 1, and then you're going to get another cytochrome C, and you're going to get another, and you're going to reduce a copper. You're going to reduce some more cytochromes, right? And eventually those electrons are going to go in. And what are we going to do to this oxygen? We're going to reduce this oxygen into water. And when we do that, that is going to pump 2 hydrogen ions from the matrix to the inner membrane space. So how many electrons do you need to reduce one O2 molecule? 4, right? That's why we do this as a 1/2, right? So you actually need, you need 4, but the way that we draw it, starting from 1 molecule of NADH, that's why we have this funky, weird, I always hate when they do that, but it makes sense, but I hate when they do that. Okay, what time is it? What do you want? Okay. So when we talk about pumping these hydrogen ions, what are the 2 things that creates for us? A change in pH and a change in electrical potential, right? That's what these 2 symbols are, right? So this is a form of stored energy, right? We have energy stored inside of that gradient, that biochemical, electrochemical gradient. And so we call that free energy. And we can represent that free energy using this equation. So we always, when we talk about energy, we talk about delta G, right? So all of the equations that we've looked at so far for this, I don't know, for the last 2 months, right, have always related back to delta G. And that's what you always kind of have to think about. So when you're trying to calculate delta G, when we looked at those those potential energies, right, of all those electron carrying molecules, if you look at that table, what you're going to notice is that it doesn't perfectly increase every time you go from one to the next. And the reason that can be different is because all of this relates back to delta G. And remember that delta G is not just a mathematical formula, but it also takes into account the concentration of our different substrates and enzymes within the membrane, right? So if you have more or less of something, is that going to change delta G? Absolutely. If you change your concentration of substrate, you're going to change delta G. So the same thing is true when we talk about reduction potential, and then that reduction potential relates to our electrochemical gradient. So you just kind of have to keep that in mind. It's all interrelated. It's all connected. And isn't this a pretty picture of all the different complexes? It's a little bit nicer than big blobs and things like that. It actually makes you think, oh, there really is a protein there, right? So we can actually calculate how efficiently the electron transport chain will convert that redox energy to a proton motive force. And I think I'm going to stop there and not do any math in the last minute that I have, right? So next time is our last class. I'm so sad. But then we have our exam Thursday. So you're studying, right? Okay.
I gotta study.
Yes, please study. Please study. All right.
Back to
The prosthetic group. Okay. Because you're calculating what you need. So you're not going to be setting my curves.
I might. Depends how I feel this week.
You should study. You should study. Are you thinking about taking biochem too?
I love you as a professor, but I do not like biology.
I can understand and appreciate that, but you've been really fun to have in class. All right. Okay, what about this guy?
One more back. No, you wrote something up on the board about like the different vitamins, the prosthetic group and vitamins.
Oh, wait, here.
Yeah.
This one.
Laboratory. You just want like all of the regular, like the methods and the materials and like all of that and like the steps and everything.
Yeah.
And then you just want us to make the Excel document as like our all of like the results and everything.
Well,
No, we need to write the results.
well, yeah. So you have to give me one example calculation,
Of course.
right? But you don't have to do tables. Your tables are inside of Excel. You don't have to do graphs. Your graphs are inside of Excel, right? So you have to show me example calculations. But you can do that. You can do that like you do your normal lab report. You can put results in data table, see Excel spreadsheet, right? And then calculations, number one, show me one example calculation, the rest are in Excel.
Gotcha.
You know.
Okay, and then that's it?
And then you have 5 questions.
Okay.
So we don't have
And a conclusion.
Oh, okay. I was about to say
Like a conclusion.
Okay.
You
Yeah,
know.
of course, I didn't conclude.
Yeah. Yeah, but that's it.
Okay.
Just fun of hair.
Yeah.
Okay. Because I haven't been able to start on it yet because I've been so busy, but
Yeah, not due till next Tuesday.
yeah.
So you got time.
Question for you.
Yeah.
So since the NAD pathway is separate from the FAD pathway.
It's not really separate. It's all feeding into Q.
Right. But I was wondering if something happens to where only complex 2 is blocked, can you still produce ATP, but it's just going to be hindered?
Yeah, but you can't. So what you can't do, that's a great question. Let me come over here.
Because whenever I was doing research for like different diseases for molecular cell, I read something about like some type of disease like this.
Yeah. So what happens if you block complex 2? Complex 2 gets FAD from where?
The citrate side.
can be the citrate cycle,
Yeah, citrate
Can
cycle
be
from fatty acids. or-
oxidative phosphorylation of fatty acids, right? And it also can be from the shuttle
Right.
system. So if you inhibit FADH2, complex 2, then you can't go on a low carb diet.
You can't, period.
No,
Oh, yeah, because you can't, because isn't any
because if you're not getting NADH from,
the carbs, like from glucose, you'd be getting the FAD from other places.
correct,
So the only way that you can actually maintain the amount of ATP that you have with a disease that inhibits complex 2 is if you eat a lot of carbs.
correct. I mean, like it's not completely exclusive. It's not completely that way, but you will be not well.
Okay.
You will be not well. We'll put it that way.
My general question.
Yeah.
What causes the electrons to bind to ubiquitin? Like, is it?
Look,
Increase in reduction.
exactly,
that's
exactly.
it,
Watch,
just
look.
increase your reduction.
That's all this is. That's all this is. Whoa,
So
come
it
back.
doesn't have to do with
Right?
like the hydrogens binding to it, or it's just based
No,
on
that's
reduction?
facilitated. It's based on this, right? So you're at Q, right? Where do you want to go from Q
Go from Q to complex 3.
to complex 3? So look what happens to the potentials. We're getting more and more positive. And these are standard, right?
Okay.
And this is not truly what happens in the cell, right? It depends on the concentration. So you have different concentrations of these different things in the list, which will change the actual values.
So it's just facilitated by reduction.
Exactly.
I don't find that chapter 11 that bad. I don't really feel like it's that bad.
It's a lot easier than.
It's definitely the easiest. I think it might be one of the easier chapters. Yeah. Maybe not like chapter 1. Chapter 1 is pretty easy.
Honestly, going back through it.
Dude, if you
Okay,
have
let me
it
close this up.
separated
Oh,
by- This evening?
yes.
Okay.
I feel like all the